Okay, and 13A, this is now early metaphase. Now, in the exam, if you have to differentiate between early, late, whatever, you do need to know those steps, okay? So make sure everybody take a picture of this and really knows it. Okay, then B, this is anaphase, right? So the chromatids are going to opposite poles. Okay, you start to see the cleavage furrow. 14. So this is now. So remember, this whole thing is showing you. So first of all, this is called what? This is in the male. What is this called? This whole process. No, it's meiosis, but it has a specific name for male. Very good, spermatogenesis. Okay, and then this is oogenesis. So here we're seeing meiosis 1 happening, and then you end up with a secondary spermatocyte. Okay, and then these two will divide to make 2 other cells, and this is now meiosis 2. Okay, so 14. We don't care about that. Okay, so then you end up after meiosis 2. From the secondary spermatocyte, you will end up with 4 spermatids. And then these spermatids will then undergo spermiogenesis to then become mature sperm cells, or spermatozoa. Okay? All right, so then oogenesis, you start off with your primary oocyte, and then it will start undergoing meiosis 1. So they describe this as metaphase 1. All right, so here again you have the spindle fibrils. Okay, and in 19 here, so remember, after meiosis 1, you will have a secondary oocyte, and then you will remove the first polar body, right? Because this cell is going to end up taking most of the cytoplasm. Okay. And then the secondary, then these two will undergo meiosis 2, right? And you will end up with another the second polar body, and then again the secondary oocyte. And here you have the other 2 polar bodies. Now they may divide, they may not, who knows? Okay? And the result, remember, is a secondary oocyte, and only if fertilization occurs will you have a mature ovum. Okay? So that's this plaque. Now this one's crazy and just too much. Where's my key?
Turn this on.
Who has my key? Yes, it is. Okay. All right, so these are showing you the different phases of meiosis. Okay? And there's a lot of information here that I really don't care about. Most importantly is, of course, what the different phases are. Okay? So under normal functioning cells are arrested in interphase. Okay? This is when the cells are metabolically active. Remember, the chromosomes are not dense. It's actually loose chromatin at this point. Okay, you have your nuclear membrane with your nuclear pores, whatever. So here you're just seeing all the normal metabolic things. So here you have your centrioles, here you have your mitochondria, your ER, Golgi apparatus with the Golgi vesicles, microtubules. The microtubules, they show as these little blue lines. No, well, yeah, the little blue lines. Here are your centrioles, the nucleolus, the nucleus, the nuclear membrane. All right, so then you start prophase. So now the cell is saying, all right, time to divide, okay? So in prophase, this chromatin, this loose chromatin starts to condense, okay? And then the nuclear membrane starts to disappear, and the centrioles come together. All right. So now we're still in prophase, right? So we continue on to prophase, and you start to see the chromosomes forming more. The centrioles are going starting to go to opposite poles, right? Okay. And we continue on. So all of these were still in prophase, okay? And all of this, their top we're still in prophase. You just see the continuation. The spindles are going to opposite poles. You keep on getting more condensation of the chromosomes. Okay, now in prophase 1 is when you start to see the chromosomes are going to start to synapse together and form that chiasmata. Okay? Okay, so as the chromosomes get together, the chromatids, okay, then what you're going to have is this. Okay, right here, this is synapsis.
Synapsis.
But this area right here is also known as the chiasmata. And actually, I don't even know if I spelled it right. Okay, so that's what this is showing you, although it's terrible. Okay, sorry. All right. All right, so then we come here and we have metaphase. So remember, metaphase is the lining up of this tetrad. Oh, and this whole entire thing is a tetrad. Okay, so you have tetrad formation. Remember, we're still in meiosis 1. Still in meiosis 1, the metaphase 1. So over here is where DNA replication occurred. Here you have crossover, also known as homologous recombination of like, let's say these strands, you have homologous recombination. All right. So then here, what do you need to know? So these are called sister paired chromatids. Okay, or sister chromatid pairs. Okay. Here we can see I really can't see the centromeres. Yeah, the centrioles. All right. And then this right here is known as or this right here, when they connect to the chiasmata. This is the chiasmata bridge. All right, so now we're getting into anaphase 1, okay? We're still in meiosis 1, okay? So in anaphase, now these sister chromatids are going to opposite poles. Opposite poles, you start to see the cleavage farrow occur. All right, and then in metaphase 2, here you have now division of the cytoplasm. You have the formation of the nuclear membrane again. All right, here you can see already that the homologous combination occurred, right, a long time ago. All right? So then you're now going to enter, once you have these two cells formed, so let's pretend this is spermatogenesis, okay? So this would be your secondary spermatocytes, right? And so now they're going to undergo meiosis 2. So here nothing is replicated, you're just having regular cell division. All right? So then this is, so now from here, you're skipping prophase 2, you're skipping metaphase 2, and you're going right into anaphase. Okay? So these chromatids are now splitting up and going to opposite poles. So this is anaphase. And then this is the end of telophase 2, okay, where you notice that you end up with 4 genetically unique cells. See that? All the chromatids are all different. Okay? All right. So, whoever wants it.